Welcome back for the last part of our genomic variant analysis part of the course. In this section, we're going to talk about how do we annotate the variants that we called in the first two sections of GVA. So just by way of a little bit of recap, the standard workflow for genome variant analysis looks something like this, where our first step was to do some quality control on the reads. The software we used was FastQC. The input files were FastQ files. We used Illumina, but you might have other read types of files. It doesn't matter. You're going to do a quality control step first in any case. The purpose of that is to trim either low quality reads or low quality base calls from the sequencer. And then the result was new FastQ files that contained the trimmed reads. After that, we did read mapping. So that's where we used Bowtie 2. There are lots of other mapping softwares. BWA is another common one. The input for that was our trimmed FastQ files, or when I asked you to do the untrimmed reads, the untrimmed, so the raw FastQ files. And then the purpose of that is to map those reads to the reference genome. The result was a SAM or a BAM file. And then from that, we did variant calling. So we used the MPileup program in SAM tools, but there are many of these. MPileup is sort of the simplest one of the simplest ones, but there's one called Freebase that I'm going to go into its strategy in a little more detail, but there's also one GATK, which is similar to Freebase at some level, and there are many more. The input here is the BAM file that was our result of read mapping. The purpose here is to identify the variants relative to the reference. Italicized reference here, right? Because whenever we're mapping, we're only identifying things that are different than whatever our reference genome is. And the result of that was a VCF file, a variant call file. So I'm providing a link here because the whole area of genomics involved in variant calling is a little deeper than we can get into, but there was a publication last year that benchmarked a bunch of these variant callers. And so I'm just giving you the link to that paper because it does a really nice job of comparing them and talking about why different settings matter, and especially when you're dealing with human variants, this is an important area, right? Because you want to be able to identify all the true variants, and you definitely don't want to have false positives, right? Basically tell someone that they have a mutation that potentially has some clinical relevance when in fact that's an error. It just wasn't called correctly. Just one word, so GATK, the one that I mentioned here, is really a software suite, not just a variant caller. It is becoming one of the most widely used, but it's so comprehensive that it's just a little too much for us to bite off for this course. But I've given you links. It has its own variant caller. It has basically all the steps that we have done and will do built into one umbrella package, which is the Genome Analysis Toolkit. Just wanted to bring that to your attention. Why is variant calling so difficult? We sort of glossed over this in the part 2 where we did variant calling with MPileup, partly because I just wanted to teach you the mechanics of how it works. This cartoon example at the top gives the reference genome and then 5 reads that line up to that. And I've highlighted a position here where it looks like maybe there's this individual is heterozygous because it's got an A in one of the reads at that position. So in principle, this individual could be a heterozygote, or that A could be a mistake. So early variant callers and simple ones still, like SnpEff, they use a relatively naive procedure that just says above or below a certain threshold % of the reads. If it's above that, so in this, the default setting is 20%, say. If it's less than 20% of the reads, ignore it. Don't call it a heterozygote. Okay, that's basically saying that is a mistake. That doesn't actually. This read should not have been A. It was a mistake. Okay, and so therefore this individual is not heterozygous. That's if the A was below 20%. 80% is just the flip side of that, right? So if it's greater than 80% G, then it's the same thing as less than 20% A. So in this example, only 1 out of 5 reads, so it's 20%, and so you might call that a heterozygote, but you don't want to say you have a lot of confidence in that because it's only one read. We cannot be certain that that is a true variant. It could be, but it could also be some kind of experimental artifact that was introduced at library prep, or sequencing error, right? The base calling error. Or it could be an analysis error where this read actually shouldn't have mapped to this location. There's another very similar location. And again, this is a cartoon example, but hopefully you can imagine if there's a very similar region someplace else, the variant, might line up with A relative to the reference and not look like a difference at this position. It might look at like a difference at some other position in that read. So a misalignment can also give you that kind of uncertainty. So this example is Freebase, but again, lots of variant calling software has gotten sort of sophisticated in the kinds of uncertainty it can incorporate into the variant calling. So Freebase, the name sort of implies the rationale here. We're taking Bayesian statistics and applying it to variant calling. If you're not familiar with Bayesian statistics, that's okay. You don't have to understand the math behind this or the statistics behind this to really sort of appreciate on a conceptual level what's going to be happening here. So translating Bayes' theorem to genomics terms, this is saying what is the probability of having a genotype, because that's what we want to know when we're doing variant calling. What is the genotype of the individual at the given position? What's the probability of being a heterozygote at this position, essentially, given the sequences at that line up on that position, right? So Bayes' theorem suggests that that is the probability of the sequencing reads given the genotype times the probability of the genotype at that position divided by the probability of getting those sequencing reads. So because in any given calculation of this value, the reads are fixed, right? We sort of know what those are. PS, we just treat that as a constant. So we can sort of eliminate the denominator here. And so you can rewrite that as the probability of the genotype given the sequencing is this probability of sequence given genotype times probability of genotype. And so then just in English, the probability, the PS given G is the probability of the reads given the actual genotypes. Like what's the likelihood of getting those reads given the actual genotypes, right? You're saying, well, we don't know the actual genotypes. Well, so what's the probability of the genotypes? The probability of the genotypes is in Bayesian statistics, it's the prior. right? So what's the prior probability? And I'll show you, we can use Hardy-Weinberg, or we can use known population frequencies to get a probability for any given genotype at a certain position. So that first term, the probability of the sequence given the genotype, the way to think about that is, what's the probability that this read is actually mapped in the right spot? Right? What's the probability of the read given the genotypes in that at that location? When you map a read to the genome, actually, let me back up a step. So just, you can read this for yourself, but just because a read is mapped to a certain location, that it's the best spot for it, but it might not be a very good spot for it. And so, right, this tells us something about the probability of, or the confidence that we should have in the genotype calls from that read, right? Imagine if most of the sites say, say not most, but let's say a bunch of these sites actually are variant, you start to say if there are lots of mismatches here, then maybe that read shouldn't have mapped in that location. So, and in any case, we probably shouldn't put a lot of confidence in any of the genotype calls that are based on that read. So that's what this is trying to get at. So that read, if it's got a lot of variance in it, it's probably, you don't want to put a lot of confidence in the genotyping calls. In the simplest case, the probability that a base pair at each position, and so this is S sub I, so that's each position in the read that's aligned to that position. You can just think of that as, what's the quality score of that base call? So if you remember back to the FASTQ files, each base pair has that bottom line that is the ASCII character telling you the quality score. We also saw that in the FastQC when we did the quality control step before read mapping. So that's the first pass, like sort of the naive probability of having that base call be correct. If it has a low quality score, then you don't want to put a lot of confidence in calling a genotype there. So it's, the confidence in your sequence call is going to inform your genotype call. Newer variant callers like Freebase and GATK can account for lots of other sources of uncertainty. So the sequencing technologies have their own error profiles. Homopolymer runs, for example, where you have like AAAAAA, those tend to be more difficult for, say, Illumina to read through than some of the other technologies. You can also have regional sequence context accounted for if you're in an AT rich region or a GC rich region, right? So if you're in a highly GC rich region and you've got one read that is an A or a T, then you might want to say that's not so probable. Okay. Also indels, right? If you're in a region where there seems to be structural variation, you might want to say indels are not so unlikely. You get the idea. You can build in additional probabilities for seeing a certain kind of allele or a certain type of variant. Okay. The other thing that we need to compute is the probability of the genotype. Okay, if you have individual samples, the way this is typically done, especially in well-studied organisms like humans or mouse or fly, is that you go to a database. If you're not in a well-studied organism, you can just say that all of the genotypes have equal probability, right? So whether you're homozygote for one allele or homozygote for the other, or heterozygote, those could all, you could say they're all 33, one 1/3 probability. But a more accurate way, like in humans, this example is for the sickle cell, or the most common sickle cell mutation, is you go to a database like dbSNP. So this is at the NCBI dbSNP website. Just took a screenshot. So this tells you the position. This tells you the allele. So T is greater than A, C, or G. So T is the reference. But there are the most common alternative to T is A, and so this is giving you information from 3 different studies, and then there are a bunch more studies. That's a link. You could go to 10 more studies, and the number of times A was seen out of the total number of samples, and then that's giving you, whoops, the allele frequency. So here you have data external to your experiment telling you what's the probability of seeing an A at that site. So that informs whether it's more likely to see a TT homozygote or a TA heterozygote, or very unlikely to see an AA homozygote. Okay. It can get even more finely detailed though. So for example, in human, where there have been lots of studies, you can account for variation among populations. So in this case, right, we've got 2 Latin American populations. They have different allele frequencies. So here, this population does not, at least in the 626 individuals that have been sampled so far, there are no A alleles, right? And so if you sequence someone from this population and your VCF file suggests that there's an A there. Sorry, not your VCF file. This is generating the VCF file. Your variant caller should say there's a very low probability of getting an A at this position. So it's very, very unlikely to be a homozygote for A, and even, right, it's in some sense impossible, given what we know, that you could be a heterozygote even. Okay, but if there's lots of evidence, right, so how would you call a rare heterozygote in this population? You would have to have enough reads, like 50% of your reads be A and the other 50% be T, to have high confidence that there actually should be calling an a heterozygote genotype at that locus. Okay. If you have multiple samples, you don't even have to rely on the database. You can get accurate genotyping by just calculating the Hardy-Weinberg probabilities from the reads that you have. So you, in this example, have 95% T at a position and 5% A. So your allele frequency is 95% and 5%. Hardy-Weinberg probabilities then, I just computed them here, P squared, 2PQ, and Q squared. Okay, so those are my a priori probabilities of each of the genotypes in my sample. Haven't even had to rely on dbSNP, for example. So this is an application of this probability, even if you're on an organism that's never been sequenced before, but you have data for multiple individuals, right? And so this is saying that it should be very unlikely. Your probability of the AA genotype is, right, 0.0025. So only 2 in every 1,000 individuals you expect to be an AA homozygote. Okay, and lots of the modern variant colors can account for this kind of stuff. Okay. The other factor, and I've mentioned regional context before, and so one of the ways this applies are when the variant colors are what we call haplotype aware. And so it's not just looking at single positions, it's looking regionally or in windows. So here I've got two examples why that matters. If you look at the window, so the top here is the reference, this haplotype, which is the reference haplotype, occurs 8 times. The T and the G mutations occur together 9 times. And then we have a couple really rare haplotypes. And so you could weight the probability of the genotype at each of these individual positions based on the frequency of those haplotypes. Where that becomes really important are in these sort of structural or indel variants, where it's not exactly clear how to line up all these multiple A's. And so what you do is say, there's an indel polymorphism here. There are 10 haplotypes that have 7 A's and seven instances where there are 6 A's. So they're basically probably 2 haplotypes in this population that vary in the number of A's, and they differ by 1. So there's a 1 base pair deletion of A that's segregating in this population. And then either, whether you have 5 copies or 8 copies, those are very rare, and so you would want to say that's a low probability. And okay. So given all those potential sources of variation, hopefully you have a better idea for why variant callers are pretty sophisticated. But it still leaves this question in sort of real terms or experimental terms of how much sequencing do you need to confidently call all of the variants. So just in case this it wasn't clear from what we did in the mapping and the variant calling already, when you map, so this diagram, right, the blue line at the bottom is the reference, and then each of these pink blocks is an individual read. And so you see across the genome, you have variation in the coverage. So you can think of how much of the genome is covered as the breadth of coverage. So did I sequence the whole thing? And then the number of times that you sequence any individual position is the depth of coverage. Right, so at this position, I have 5x coverage. But you can see, like out here, I've only got 3x coverage. And so how deep do you have to sequence to get, or how much sequencing do you have to do to cover everything? So for the human genome, this has been probably the best studied. This is from a paper where they did 10,000 genomes. They did this in replicate, so it's not 10,000 individuals. You can go read the paper for the details, but the main point is summarized in this graphic. The blue dots are the proportion of sites that are actually covered at 10x. The orange are the proportion of, it's the proportion of the genome that is covered at 30x. So if my target was 30x coverage, so I get an average of 30x coverage, only about 60% of the genome actually gets covered at 30x. So you need to let that sink in, because this is an important thing to remember when you're doing genome sequencing experiments. If you only targeted 7x, which before this paper was sort of the rule of thumb, you should do 7x on resequencing. Well, that means that less than 50%, it's actually down here somewhere in the 25 to 30% range, is covered at 10x. And if, right, thinking about this, how would I become more confident in that being an A? Well, part of it, I could get the database and see what's the allele frequency at this in the population, or I could, double my read count. And if it's real, I should see A pop up a couple more times. What this study said is that if you need to get it, like to call all of the variants in the human genome, at 10x, you have to do average 30x. So just to reiterate, because I know these numbers can be confusing. To be confident in every true variant call in the human genome, I need average coverage of 30x, because what that actually gives me is the proportion of true variants are all covered at about 10x. So at 10x, I can be more or less confident that I'm going to get a real, have the statistical power to call that variant. Okay. So that I highlighted this was the take-home message from this paper, is that you need to, if you want to get every variant in the genome, you need to target at least 30x average coverage. The second thing is that you don't get much return on investment by going deeper. So, right, you have a very shallow slope as you get out here to 50x, right? You're barely a few percentage better in terms of the number of true variants that you can call. Okay, so where are we? We're trying to get down here. We've done quality control, we've done read mapping, we've done variant calling. Now we want to figure out what are those variants, what do they do? So we're going to use SnpEff on Galaxy. I'm giving you the variant call file. Purpose here, like I said, we're going to annotate the variants, and then the results of this step are statistics, and in some cases we'll see some pretty simple visualizations. So the background for the tutorial data, where does it come from? This is based on a short read sequencing, so Illumina, from an exome. So they used exome capture on chromosome 22. So this little asterisk you can see down here, they had oligo probes to pull down exome from RNA. Okay, so you know, probes that matched the genes on chromosome 22. So they pulled those down out of whole genome DNA. This is from the most sequenced individual on the planet, probably. She's been in all the big genome projects in the US. Her quote unquote name is NA12878. Sounds like a stormtrooper name, but that's the data that we're working on with. Like I said, it's based on a whole genome tissue extraction. These are things that might be relevant, say, for some variant callers, but the kind of prep, because like I said, you can account for different error profiles in library prep and in the sequencing platform. But in this case, I've already done the quality control mapping and variant calling for you. We're just going to look at this for the annotation part. Okay. And even though this was a exome pull down experiment, there's always genomic contamination in the final sample because that enrichment process, the pull down, isn't perfect. So we'll probably see some sites, for example, that are not on chromosome 22. Okay, so how do we do this? First step is to create an account on a software platform called Galaxy. So I've given you a link here. See if this works. Yes. Okay. So that should take you to a website like this. You will go here to log in or register, and it's going to ask you to put in a public name or email address. It doesn't matter. This does not have to necessarily be your UTA email address. This is for you to be able to log into Galaxy. I will not have access to this. I don't need access to this. I'll show you how to share it with me at the end. Okay. So that's the first step. Create your account on Galaxy. Second step, create a new history for this tutorial. So once you create an account, you should be logged in, and you'll probably have a blank history like this if this is your first time on Galaxy. And then you'll hit the plus sign there. If that's grayed out, you've already got a blank history here, and you can just click on the pen icon, which is what I'm showing you here, to rename that thing. So let me just show you how that works. I can't do it on this brand new. Log myself in here. Okay, so right, you'll be at a spot like this. You won't have SnpEff already ran, but you can rename this by clicking on that pen. And I want you specifically to name this with your UTA ID number. Okay, so for example, for me, it's 1000-585920, something like that. But the name of your history should be your UTA ID number. And I need it to be that so that to just make it simpler for me to grade which history came from which student. Okay. So at this point, right, we're right here, click on the pencil. Your unnamed history should become your UTA ID number. So where it says copy of 2023 Bioinfo Dameth, yours will say your ID number at this point. Then the next step is to navigate your browser to that. Whoops. Let's go back to this link. And this should bring up a page like this. And for me, it's saying switch to this history, but for you, actually, I'll log out. Just so you see. Oh, it's not working. Okay, so if you paste in that URL, it should, hopefully for you, it linked straight to it. Because for me, because I'm logged in as me, it is not letting me. There. So when I logged out, which since you won't be logged in as me, you'll see this icon to import this history. And that will give you an opportunity to import that into your history. Get back in myself. And just want to show you what it should look like. Right, so your Galaxy instance should look like this, where all you have is something called VCF file, and that's imported. So that's just me sharing the VCF file with you. So like I said, I've already done all these steps. I have the VCF output, and then I'm sharing that VCF through Galaxy. Okay, so if you followed those steps, you should have something that looks like this. Just a quick note about Galaxy, because this is probably the first time most of you have seen it. It's really just a browser-based interface for a lot of bioinformatics software. This whole left-hand side is a toolkit for all kinds of genomic analysis and transcriptomic analysis and protein analysis. Well, so besides just being a graphical interface, it also is recording the steps. So you'll see as we do things, it'll build up in our history, and then you can publish that as a workflow in a publication so that people can copy your analysis or recapitulate your analysis exactly the way you did it. So this was a big step forward in the early days of bioinformatics about making analysis transparent. It's just point and click. You could import somebody's whole history and see exactly how they did their analysis. The main instance of Galaxy that we're using, it's actually doing most of the computing on HPC systems at the Pittsburgh Supercomputing Center. Some of it will be done on bridges. All of that is going on in the background, and you don't have to worry about submitting blast jobs to the Slurm queue. It just does it for you. Super handy. Another great thing is that you can get the Galaxy software, so you can get an instance just like this downloaded from this website and put that on your local Linux machine if you wanted to. Now, that's not trivial to get up and running, so I would say don't just go download it and expect it to run. It takes an investment, but it is a worthwhile investment if your lab does a lot of genomics. So that's all I'm going to say about Galaxy. Awesome tool. So once we have our VCF imported, right, we're here. If you click on this little I, you'll switch over. This middle column now is our VCF file, and you can see the header on this one is much longer than the E. coli header that we have. That's because in the reference genome sequence for human, there are the main chromosomes, so up at the top. So for example, is chromosome 1 and then the length, but then it's got all the individual contigs and alternate genomic structural variants. So that header is really long before you get down to the actual variants themselves. Right? So this bottom section looks similar to the one that we had for E. coli, where it's giving you chromosome, position, strand ID, the reference allele, the alternate allele, and so on. And this scrolls to the side, right? So just like our other VCF file. We could have done the E. coli one, right? I could have done this as a continuation of the second part where we did our own variant calling. I just thought it would be more interesting to work on human. That's all. The SnpEff results you might find more interesting if you know they're from human. So that's the only reason to switch over and give you a different VCF to work on. Just a little background, a couple links again to variant call, like if you want to know what's the information that's actually in that file. And then really the reason we're here, let's annotate those things and see what they do. So on the left-hand side, you should search for the SnpEff tool. So here over on the left of this window, type in SnpEff and you'll get these options pop up. We're going to use this one called SnpEff EFF. Right, so right there on the slides. And you can see it automatically populated. We only have one VCF file, so it automatically populated that. And then in the genome source section, you want a locally installed SnpEff database. And then you want to pick HG38. By default, it's picking 38.86. We want to go down and pick number 38. And then we're going to leave everything else as the default. If you scroll all the way to the bottom, you can see additional detail about what SnpEff does. It gives a citation to the paper. There's another tutorial that you can run through if you want to. This is the paper that describes SnpEff. But you can leave all of those default and go all the way down to run the tool, and then you hit run. You saw they were gray, and then they were gray when they were queued, and then they were orange when they started to run, and they'll stay orange until they finish, and then they'll turn green. So I showed that here, right? They're gray when the job is submitted, then they'll turn this orange or yellowish color while they're running, and then they'll turn green when those two outputs are done. You can ignore the job numbers here. I don't even remember. I must have erased a step. So it doesn't matter whether you're, and if you have, like if something doesn't run for some reason, it will turn red if there's an error. In that case, you can try to make the adjustment and then run the tool again until it completes successfully, in which case it will turn green. So of those two outputs, one is a VCF formatted file, looks very much like the input VCF file, and then the second one is the HTML, which is the one we're really interested in. That may take a while, so I'm actually going to switch over. To whoops, that's not what I want. This. All right. I'm just switching over to a different history where it was already finished. Actually, I saw that it had finished right before I did that, but that's okay. And so if you click on the I for the HTML output, you should get this middle section. And so this is the SnpEff summary, and then this is hyperlinked to the different sections of what's in, all of the annotation that it's giving you about the SNPs in your data. So how many variants were there, the number of effects of those variants that it predicted. So it's looking at, where were the mutations? Did they cause an amino acid change? It even considers whether it changed the type of amino acid, like from hydrophobic to hydrophilic. That would be a more not dangerous, potentially more deleterious, right? If it changes the character of the amino acid out of position. Here you can see the rate across the genome. Remember, I mentioned this earlier, that this was a pull-down experiment for exons on chromosome 22, which that's where most of the variants we found are, but clearly it was imperfect, right? Because we found variants on some of the other chromosomes. These other contigs are structural variations, so things that are not in the sort of canonical chromosome 22. That's what these two are. The duplications that are on chromosome 22. So here, rather than walk completely through this, right? We have SNP, we have multiple, I think I have this in the slides, right? So multiple nucleotide polymorphism. So insertions, deletions, mixed. So that's multiple nucleotide and indel, so complex mutations or variants. The impact, so there are 136 that are predicted to have a high impact, right? That's typically strongly deleterious, right? So those would be like a premature stop codon or something like that. It's likely to completely knock out the protein, for example, or to knock out its function. Missense, nonsense, silent. I'll let you scroll through these, but so you'll have to look through these to answer some of the questions at the end. Right. So last slide, the homework due Tuesday the 28th is to complete the GVA quiz on Canvas. The main part of that, well, one part of it is to share your completed history. So the same way I shared the variant call file with you, you're going to have to share your result with me. So you click this little arrow and then come down to share or publish. So that's 1 and 2 on the slide. You'll get this, click that, and then you're going to copy the URL and you will paste that. So this is where it's important that your UTA ID is the name of the history, because that's going to affect the URL here. You should see your UTA ID over here, and then that will be built into the URL. And so that will tell me, that will put a history that's uniquely linked to your ID. And so you're going to paste that into one of the answers on the Canvas quiz, so that I'll have access to see that you completed this whole thing. Okay. All right. So happy annotating.